So good afternoon, everybody. Thank you for joining us for the last Journal Club of the year. It's held by me, but surprisingly, it's not on metabolism. And so you can all take a deep breath and relax. The topic is something that has caught my attention, I think all of us during our call for the internal grants, where these two people, one from the Hospital Pellegrin and one from Olevec, they came to propose their microdot for autoantibodies against atrial fibrillation, and their work is based on this paper. The paper is quite short, so I hope that we can discuss a lot on it. There's a lot of nice hints. It's mostly exploratory and there's a little bit of functionality, but not too much, and it's in rodents. But I think that the originality of the topic deserved the publication, and in principle, I agree also, even if we will see that some data are missing in even in the publication. Okay. So as usual, when I present, I like to introduce the authors and the journal. This is particularly relevant this time because now Andre doesn't come from a cardiological background, so it's also important for him, I think, and for everyone also to remind ourselves which kind of journals we have to we can target and in which context. So surprisingly low number of authors. This looks like more like a communication letter other than a publication. But again, the results are quite impressive. And the people that are involved, they're fairly known in the field. They are already established scientists and established experts. And they come from Bern, Zurich. And in part, also, they contribute from Canada, from the Montreal Heart Institute. The journal, when you find this paper, is Circulation. It was published in July of this year. And so Circulation is not, like sometimes we see, is not a general purpose, high impact journal. Is very specialized in cardiology, and especially on the not so much on basic science, something that is basic science but mostly connected to clinical observations, and as you can see here, also epidemiology, health services, and outcome studies, advanced in basic and translational studies. The impact factor is not, it was not written on the webpage, so I tell you it's 38.8 this year. So, and it's still growing. Okay, now coming to the topic of the paper. So many of you know that in the general population, the prevalence of atrial fibrillation can be in the order of 3%. So it's a quite large amount of people that are affected by this condition and by its consequences, also in terms of risk factors and hospitalization rate. But there is usually in the large majority of the cases, atrial fibrillation is accompanied by other diseases or comorbidities or other conditions. Most recently, metabolic syndrome has climbed the chart among the conditions that are associated with atrial fibrillation, for example. But a small amount of around 3%, between 3 and 5%, as is shown here, of atrial fibrillations is not, it's called unexplained. In the past, it was called lone AF. It's atrial fibrillation that appears in sectors of the population without an apparent comorbidity or in very rare cases, not even in the normal um, risk range for due to aging. So the etiology of this lone or unexplained atrial fibrillation, it's uh, it's quite a head scratch because sometimes we associate it with atrial myopathy, secondary to heart failure, or to ischemic heart diseases, to CAD. But um, we and now I spoiler the rationale of the paper. We rarely associate it with autoimmune diseases. And if you see here, for example, I put um, for you a 2020 update on the cardiovascular diseases um, um, risk factors. And the only one that is relatively associated with autoimmune diseases is rheumatic heart disease. So something that is correlated with, for example, rheumatoid arthritis or psoriasis. And so it's even in 2020, it was characterized as 1.6%, even secondary endocarditis 0.4%. So, but if we look a little bit further, sorry, move the panel of people so I can see. If we look a little bit further, in this study published in The Lancet 1 year ago, for example, we see that if we look at a large cohort of the population, in this case, the UK Biobank, and we look at people who have diagnosed autoimmune diseases, we see that there is a significant increase in the risk of developing cardiovascular diseases. The follow-up here is less than 2 years, but in the study can go up to 5 years. So it's around 1.5 the hazard ratio of the matched control set. If we go a little bit deeper, in particular for atrial fibrillation, atrial flutter, we find as well an increased hazard ratio in the cohort that presents with autoimmune diseases. The question arises: does some form of autoimmune diseases can explain this lone atrial fibrillation? And this is what the authors of this paper are investigating. So there are two biobanks that are pooled together in this study in order to study these autoantibodies in the plasma of this of patients or of screened individuals. So there is this Discovery Life Science Biobank in Switzerland, and there is this other biobank which is not really described in Montreal. The patients are classified based on the ICD-10 code. There's more details in the paper, but it's basically three codes that assign them as diagnosed atrial fibrillation or not diagnosed atrial fibrillation, hospitalized, but without a code for atrial fibrillation. It's in total is a cohort of 88 individuals. From Montreal, we have this 14 pre-AF cohort, which means that these individuals, they were sampled. They did not present with atrial fibrillation, but in the 5-year span of the follow-up, they developed atrial fibrillation. So they are using this cohort to see if there is a pre-existing autoimmune condition that can underlie the development of atrial fibrillation in time. The largest set is in this, which what I call this diagnosed subcohort. So we have unexplained or lone AF, 37 patients, and they matched patients without these codes via age and sex, and they matched it in a one-to-one. Here on the right, you can see the demographic of the cohort. Um, there are no there are no statistically significant difference, as one hopes to see, not to have any bias. And the authors present the complete design of the study, but we will go through it as we look at the experiments. Basically, once they describe this cohort, they design a peptide microarray against which they test the plasma of these patients or the controls. And then they do some biostatistics for the relative expression of certain types of autoantibodies against ion channels. In particular, you will see that they focus on KCNJ5, KIR 3.4 and KIR 3.1, so the potassium acetylcholine current. And once they have characterized this signature of these autoantibodies, they isolate them with affinity chromatography. They expose on the one hand IPS-derived atrial cardiomyocytes, and they perform patch clamp and carbachol response. We will see it later in the detail. And then they will perform mice immunization or sham immunization. They will look at functional mechanics with thoracic echo, and they will do also ECG with the classical stress test that we do here as well to try to see vulnerability to atrial fibrillation. So this chip, this pepper chip, is a custom chip that you can order from, for example, for this company. This is something that we discussed also internally, for example, during the last Horizon Europe call, because they are very powerful and they can be tuned to whatever you think your target is. So once here, they seem to have a quite narrow target. They knew that they wanted to study specific ion channels. And so they focused on, but nothing forbids, this is what I want to say, nothing forbids to use proteomics in the plasma, for example, to study what level of expressions are there for different antibodies and then design your array. So they focused on anti-M2 muscarinic acetylcholine and beta-1 adrenergic receptor antibodies. There's a short description of the dilution rate and how to incubate these samples. So it doesn't give a specific binding to the substrate. So as you can see from the table, what are the AF-specific autoantibodies that they have found using this peptide microarray? So if we look at this, if we look at types of comparisons, so AF versus control for what concerns the Swiss biobank, and pre-AF versus control, so the bank in Montreal and the bank, the match control that they had in, I think the control comes from Zurich, but I might be wrong. So the main hit that comes out, of course, there's a much larger table. In both of these comparison is KIR 3.5, 3.4, sorry. Statistically significant, suspiciously similar, but it's probably due to the FDR correction because the p-value is different. Also, the numbers are different, so I don't know how I think it's a trick of the statistics. This is the sequence that is associated with this autoantibody. So this is just also for me, because I'm not an expert of ion channels, a reminder of what KIR 3.4 and the gene that expresses it, KC and J5, kind of channel they underlie. So it's a potassium channel. It belongs on the inward rectifying potassium channels, and it's part of the muscarinic receptor. So with GIRK 1, it forms part of the muscarinic receptor. And mutations of this gene has been associated with a plethora of arrhythmic diseases. For us, it's interesting atrial fibrillation, and for you, I'm sure it's interesting also long QT. So this peptide sequence, what does it correspond to? So the antibody is targeting this part of the pore, of the extracellular part of the complex. So it will affect likely its function. And this is what they observed in their cellular studies. So the usual caveats, these are immature atrial myocytes. They are spontaneously depolarizing, and they are cultivated for a relatively short amount of time. But they come from this company, so they live to the company. The proof that these are atrial cardiomyocytes. What is important for us is that when they are subjected to carbachol, which is the agonist of the KIR 3.4 of the acetylcholine of the muscarinic receptor as a whole. We see an increase of, as expectedly, of the inward rectifying potassium currents and a shortening of the action potential. If we expose these cardiomyocytes just to the purified autoantibodies, we observe a similar effect. And if we combine the two, we can potentiate it, but not significantly. And here is the bar graph that shows basically what I explained to you just now, especially this point where the addition of these autoantibodies with carbachol doesn't significantly shorten the action potential, although like qualitatively it might seem so.
I can interject.
Please.
What is more convincing is that heart rate is going down with carbachol, and it's going up with the body.
Yeah.
Don't they notice it?
They don't report it, surprisingly. They don't report it. They just discuss as all this increase in the ACH current when they are exposed to these autoantibodies. And maybe you can comment this for me. The significance is in this range, which I would associate with an hyperpolarized state of the membrane.
Yeah, although it shows a depolarization, I think, but yeah. Well, anyway, it fits with the observation that vagal stimulation or acetylcholine slows down heart rate.
Yeah.
And if you antagonize it, it speeds up heart rate.
So we're almost at the end. Don't be sad, but we're almost at the end. The last part of this paper, as I mentioned, is this immunization trial in this in Balzi mice. So male and female in 1-to-1 ratio, randomized for randomized for sensitization with a synthetic version of this peptide. They describe the strategy of shielding the peptide with a cysteine. It's all very technical, and I got lost several times. But it's just to say, like, it's synthesized but it still maintains the sequence that they studied in the previous step. And what is important for them to mention is that, and for us as well, is that this sequence is preserved, is highly preserved between human and rodent KIR 3.5. So the animals are immunized following. They are immunized at several steps. 9 days, 13 days, and at 15 days they are they undergo the terminal experiments for echocardiography, ECG recording, and electrophysiological study. Again, there's no heart rate. Well, you can see it here on the RR interval as well. Very frustrating, as you might be familiar with. You test your model, and if you don't stress it, nothing is visible. I didn't report the transthoracic echo because they didn't find anything as a matter of mechanical function. So it looks very much similar between the sham immunized and the KIR 3.4 immunized. Yes, I don't know if is this possible to measure in human. I know it's in rodents. I'm not sure. What happened if we stress it? Well, then it's a different story. The atrial ERP significantly decreases. Sorry, this legend is not common. This is immunized and this is sham immunized. So the atrial ERP significantly decreases, which is a good hint of atrial vulnerability to rapid atrial pacing. And as you can see in these representative figures, after pacing, if they are immunized with this peptide, which resembles the autoantibody, then atrial fibrillation is triggered significantly easy. So you start from to a little bit higher than 20% of animals that are sensitive to develop episodes of atrial fibrillation to 60%.
But isn't it?
Sorry, to 80%. Please.
Yeah, how do they stress it? By pacing, I understand.
Yes, from the right atrium. It's from the jugular, but the protocol is not described.
No, but if the animals get atrial fibrillation in a certain percentage, then isn't it logical that the atrial ERP is going down as a result of the high heart rate during atrial fibrillation?
I think this is before the stress test.
Before, oh, okay.
Yeah. No, I wouldn't like. And this is confusing. Sorry, because it's the legend is missing here. This is not a common legend. So I think I remember the electrophysiological study was done before the stress test. So significantly increased percentage of vulnerability to atrial fibrillation episodes, and also, but not significant duration of atrial fibrillation, as you can see here from this compound point. Interesting. Of course, there is no substrate, and we might miss some modulations. This is very different, for example, from what we see when we try to push mitochondrial ROS. We see both an increase in atrial fibrillation episodes and increase in duration, but the mechanism is different. And as a conclusion for these authors, they claim, and we can discuss about it, the description of a novel autoimmune form of atrial fibrillation caused by KIR 3.5 autoantibodies, which seems to be restricted to these lone AF cases. But further studies are needed, and blah. And especially if we can find a way to neutralize these autoantibodies, because of course there is no therapeutic evidence that blocking these antibodies is actually working to reestablish the normal sensitivity to atrial fibrillation, at least in the models that they described. So thank you for your attention. I mentioned it was short, but I hope you enjoyed it.
Very good. I like short. So are you going to treat your patient with colchicine now, or what do you suggest that this affects the clinical practice?
I think we need to find out first if this is true. As a matter of fact, in patients, at least what the kind of patient that we have available here, so either with long-standing persistent AF and other comorbidities, or patients that have AF diagnosed for scheduled for atrial fibrillation. This is something that we don't know. We don't know if these antibodies are at a very specific are purely for this kind of potassium channels, or if they are also. I wouldn't be surprised, like if we saw, even in the table, you saw there was some hint that also L-type calcium channels autoantibodies might be significantly represented in these patients.
Yeah.
I wouldn't be surprised, because they are one of the first targets of post-translational modifications during these long-term remodeling processes. And I mean, the ratio between the significantly interesting autoantibodies might change in patients that had this arrhythmia for a long time. Actually, I don't know how. I don't think they described how long these patients were in AF.
Patients with immunosuppressive therapy after transplantations, kidney or whatever, do they have more or less atrial fibrillation?
Good question. Might be one of the cases in which the heart is the least. At least the electrophysiology of the heart is the least important thing, because you have an alien organ inside of you.
Yeah.
But I've never investigated it. I saw recently that it was described a paper about lung transplantation after resection for small lung cancer cells. And it seems to be, in this paper, there was a higher vulnerability to develop atrial fibrillation.
Okay.
It might be post-operatory in these cases, so yeah. Because it's the lung, so
yeah.
I don't know. I don't know the kidney, actually.
So do you believe it?
I am intrigued. I don't know if I believe it. It's a nice study, but it doesn't add a tremendous amount to the etiology of the disease. It's of course, because this 3%, 3 to 5% of lone AF, they target. So they're merely targeting the male sex, and they are in the range between 40 to 50 years old. Then it might be it's definitely relevant as a matter of secondary prevention, because you have or even primary prevention, because you have a known risk factor. So in this term, it's certainly useful to investigate, to reduce of 3 to 5% the cohort of patients ahead in 10 years. Is definitely interesting. But even the group that proposed the internal project, they expressed it very well. We don't know enough about patients. But this is what they wanted to do. Actually, they wanted to develop some sort of assay based on microdots, so that they could, take some serum and
Yeah.
do like this glycemic test in the hospitals. I don't know if I believe it.
Okay.
Do you believe it?
I'm a bit skeptical, especially because of the IPS cells. I know they are very unreliable.
Yeah, especially for potassium currents, I really don't trust them.
And the examples that they show action potentials with the shape of a ventricular action potential.
I know that's the same, exactly the same commentary that I have every time I see an action potential from these cells.
Yeah.
So look, we can leave we can leave it as it is, but we know among us that this is not an atrial action potential.
No. Well, but the concept, the idea is interesting.
Yeah, and it's easy to deploy. So, as a matter of fact, could, I'm still. I think they're not saying a lot about why they designed these peptides. I think there is. It smells like intellectual property here,
Ah,
because why this peptide specifically?
yeah,
Um, is it because, I mean, it's not described? If, for example, it makes sense to target the extracellular portion of the channel, because
yeah,
that's what accepts the agonist or the antagonist. Uh, but it wasn't clear for me if they broke the peptide in two and they try to look for different antibodies, because you see that it makes these loops.
yeah.
So usually the antibody would fit in one of these loops. Um, so I want to give them the benefit of the doubt and maybe have a further look at the microarray table, which is not easy to look at. Um, I want to give them the benefit of the doubt that they tried both of these loops.
Okay.
Um, oh, missing data. They mentioned that these cells present an atrial phenotype, and they say that it's in the supplement.
Ah,
But I I challenge you to find it in the supplements.
oh, okay. That's an interesting supplement.
Yes, it's not there, not at
No.
all.
So how do you explain that this ends up in circulation?
Ah, have you seen the first author? um, I think the innovation. I mean, it's not a terribly designed paper. It's just that it doesn't break the standard, but for sure it's the first description that I've seen of a preventive autoantibody.
Yeah.
So I think it passed for this reason.
Okay.
I mean, the maybe the mouse was done as a first test, but these cells, they again, they smell like reviewer question.
Yeah.
No, they, they are well made, and cutting the intermediary by buying them from somebody is a smart move when you don't master the differentiation of these cells. Also, when you don't have 3 months to mature these cells into an engineered
Yeah.
heart tissue. These cells in 1 week they are ready.
Yeah. I
You
know.
plate them.
We
They
use it as well.
yeah. So once they are synchronized,
Yeah.
they they, they will take, if you don't stress them electromechanically, they will take a year to spontaneously mature.
So are there any more questions?
I have a very kind of globally kind of dumb question, but let's say you find the antibody you think is the most important, and now all of a sudden you start getting rid of it, suppress it, block it, whatever, but there was another antibody that is actually worse and binds to the channel, right? I mean, is this a potential that anyone thinks about? Like, I mean, you can go through all of this stuff, but once you start messing with the immune system, I feel like it's way more complex than simply just finding one thing and eliminating it, and all of a sudden the AF is gone. I mean, this is something I don't really know if anybody thinks about this, because maybe it's actually bad. When you start putting it together, because the second thing, I mean, that's really no answer. That's just something that kind of perplexes me. But the thing there is an answer to: are most autoimmune diseases are they always genetic, or is this something you can develop over time as having be exposed to something as well? You know, constantly exposed to some antigens and things that develop and like you overexpress through your sympathetic. I mean, your immune system is kind of overactive. Don't know.
I will let the physicians answers to the second question. I will give you like my grain of salt on the first question that you asked. Okay, tampering, trying to inhibit this autoantibody might in the end reveal another autoantibody below. It's premature to give an answer to this because we don't know why these autoantibodies are formed. The channel might be released for many reasons, and it might be interpreted by the immune system as a dump, and so it develops antibody against it. It's not described, it's not even mentioned in the paper, like why these autoantibodies exist, or if they are secondary to an initial insult, a pre-existing insult. This is not described in this. I would expect some autoantibodies to be created as a response to a cardiovascular insult. That, for example, for atrial fibrillation, once the cells start to die and there is a replacement fibrosis or fibrofactory activation, and of course all these proteins, they go in circulation and they might trigger formation of autoantibodies. That there's a quite extensive literature mentioned in the introduction of this paper about it, but this is not what is happening here. So, is there an initial insult? I don't know. And what will happen if we tamper with it? It's certainly, it poses some dangers, but I think it's premature, just based on this paper, for me to give an answer. The second question was how do you develop these autoantibodies?
Yeah.
I think I leave it to you.
Okay. Anything else? Andre, since you are connected twice, we expect a question.
So nice, he connected twice.
No, actually, no. About regarding this, what's the main cause of the autoimmune problem? Yeah, somehow I think this both environmental and hereditary stuff. Let's say same as with cancer. Yeah, so it's some kind of predisposition, and also external factor could modify this. I have one stupid question actually. I really didn't get. These autoantibodies, they found in every patient with AF from both cohorts or in some separate patients?
Yes, in the patients that developed, either they presented with diagnosed unexplained AF, or they later in 5 years from their sampling developed AF, they found that, they found that the autoantibody against this channel, it was always significantly represented. You see here is mean pre-AF and pre-AF versus control. So either when you come with the diagnosis, or when you don't have the diagnosis but you will develop it, it seems to be a risk factor to have these antibodies, these autoantibodies. But you see, what I was mentioning is, for example, we have here 6.2. This is also significantly represented, but only maybe as a preventive risk factor. And also antibodies against CAV1.2. It's it's very significantly expressed. So, I mean, at some point, the authors wanted to tell a story, so they focused on the one that is at the cross between diagnosed AF and developed incident AF.
Yeah.
But I mean, there's a large story here. I mean, look at this p-value. Saying out loud.
Which one were you pointing at?
Oh, sorry, the last lines.
Oh, yeah.
Hey, look at the false discovery rate. It's huge. And by huge, I mean small. Miruna, do you know anything about B cells, formation of antibodies, or what is known about this kind of research?
No, I don't know.
Yeah.
You chose something outside my
But
field.
sometimes it's
Yes, very good.
Guido, can you hear me?
yeah.
Yeah, sorry, I'm talking from Andrei's microphone. That's the reason why he's connected twice, because I didn't have a microphone.
Go ahead. What do you think?
Very interesting story. I was wondering actually about this potassium channel. I mean, is it only, I'm not a cellular electrophysiologist, but is it only expressed in atrial cardiomyocytes, or do we also expect an effect on ventricular cardiomyocytes?
No, it's part of the M2 muscarinic receptor. You see, it's associated with long QT syndromes and other types of ventricular arrhythmias.
Okay, so in principle, it could also exert effects on the ventricular side?
Yeah, which is...
did they comment on this, or did they study this in any way?
No. Only the small conductance potassium channel is atrial specific, right? I speak to the audience.
Yeah.
Yeah, okay.
Well, the ACH channel also is predominantly present in the atrium.
Okay.
And
So
also,
yeah, it's this channel is predominantly expressed in the atrium, as Ruben was saying as well.
Okay.
All right.
Okay.
All right.
Thank you.